Banerjee is a research assistant at the University of Sheffield in the Cognitive Ability and Plasticity Lab. She will talk us about modeling behavioral task, which is an inhibition task, a stop signal task, and how it interacts with associative memory. So please, Nilosmita, whenever you're ready.
Yep. Can you see my screen though?
Yeah.
Perfect. Okay, cool. Right. So, hi everyone. I'm Nilosmita Banerjee. I'm currently an MSc graduate in cognitive computational neuroscience from the Department of Psychology, and I'm going to talk you through my MSc project that I've recently worked on and submitted just about a couple of months ago, and it's titled "Modeling Appetitive-Disinhibition in a Modified Stop Signal Task: A Computational Model of Associative Memory Initiated Disinhibition." Right, so today we're going to cover a bit about the introduction and the background, the research gap, and the current aims of the study. We get on to looking at the methods, the results, and then just concluded. Right, so we looked at inhibition. So inhibition is one of the most important executive function that enables us to efficiently drive around in our environment, make optimal decisions, and it's important for tackling goal-irrelevant behavior. And response inhibition has been studied for long in cognitive psychology and studied using the putative behavioral paradigm called stop signal task. Many mathematical models have been proposed in the past, but the horse race model happens to be one putative model that's been used long to quantify and predict the performance of participants' inhibitory control power in the stop signal task. So this is a schematic of a typical stop signal task, which is used in most of the cognitive psychology and also neuroscience paradigms. So just to run you through a little bit, every single black box is basically a computer screen that appears to the participants, and the arrows, as you can see, are what we call the neutral stimuli. They point in either of the directions, right or left, and the participant is supposed to press the keys designated to the directions when they're doing the task. In a stop signal trial, the participant will be given a couple of co-stimuli, which is pressing right or left, and will be interspersed with a stop signal like the cross in as shown in the schematic, when the participant is signaled to basically inhibit their prepotent action to the particular task. This is a more descriptive schematic, which also shows the trial and the participant reaction time and what constitutes of it. Right. A large body of research has demonstrated that the ability for appropriately inhibiting some goal-irrelevant response or that prepotent response terribly impairs when a participant is asked to do the same in an environment which is called appetitive stimuli or appetitive choices. And this has been captured by a body of over 30 years of research in experimental psychology studies that have used a modified version of the stop signal task, but with appetitive pictures instead of the neutral sort of the arrows that we saw in the schematic before. So this is a schematic which shows that it exactly is the same. The task is not different in its structure. The only thing that changes perhaps is the stimuli that is used instead of the neutral stimuli. They've used the appetitive stimuli. In this case, we've shown chocolates. This is actually a paradigm we've picked from, the schematic is picked from a paradigm which was used in some actual experimental psychology studies done. Right. Now, the research gap that we found was that every single model, mathematical model that was developed so far to explain the behavior was descriptive in its nature. However, the standard sort of the process models that explains the mechanisms of inhibition that is involved in a stop signal task was proposed only very recently by Bouchard and colleagues in 2007. The problem was that although this particular interactive race model explained really well what was going on in a stop signal paradigm in terms of explaining the inhibitory behavior, they did not really capture, or the parameters of the model did not really capture or explain what was happening in the impaired response inhibition phenomena that we saw in these modified stop signal tasks. So in simple words, it did not really explain what was going on when the participants were presented with an appetitive cue in a modified version of stop signal task. They were showing disinhibition, but the model did not have any parameters that could capture why this was happening or how this was happening. So that's where the aim of the current study came in. So the current study aimed to build a process mathematical model similar to the interactive race model, or what we did in this study was build on the interactive race model of appetitive cue initiated disinhibition. So we were trying to capture the disinhibition element that was observed in the modified stop signal task with the appetitive cues. And theoretical frameworks, a little bit of, because this was a very theoretical project, we tried to base all our augmentation processes on what the theory had to suggest, and the literature showed that the theoretical frameworks have indicated that such a disinhibited response or a going behavior or approach behavior towards these appetitive cues is usually an outcome of recalling these approach behaviors from one's associative memory. There's a lot of research even in neuroscience that focuses on the elevation in the level of dopamine, for example, that sort of happens when appetitive cues are presented, which has been considered to be an instantiation of associative memory recall initiated behavior or going approach. So in order to therefore address this, we built an augmented version of the interactive race model of action suppression with an associative recall free parameter, which we called lambda. Lambda instantiated the associative recall of the approach behavior in our current model. So the methods we used, we tested 3 different parameter variations for augmenting the associative recall parameter lambda in the interactive race model of action suppression. And we formalized 2 different lambda parameters, one of which was the lambda go, which instantiated the approaching behavior or the disinhibition that we observed in the participants for the modified stop signal task, and then the lambda stop, which instantiates the inhibition behavior, which is usually what is expected. So before starting to test the variations, we started with the actual model. So this is the neutral variant. We called it the neutral variant because it was well simulating the behavior that we observe in a non sort of non-appetitive condition. So it was because it was explaining that, we started off with it and we called it the neutral variant. So just to walk you through this original model a little bit, the first equation models, the parameters of the first equation models the going behavior, and DAGO basically stands for an accumulator. So we have formalized the going process as an accumulator that starts from, that races from a particular starting point, and it races towards a threshold theta. So every single parameter in the first as well as the second equation actually conceptualizes and formalizes that sort of racing between the 2 processes of pressing a button or not pressing a button, for example, in the case of a stop signal task, so going or stopping. So DA stop, the equation 2 basically formalizes the stopping process. And I just want to draw your attention to the most important part of this particular equation, which is the part of the beta parameters. If you can see, the beta stop and the beta go. These are the important parameters in the equation specifically because they talk about the interaction that the 2 processes have. So for example, if there is a go sort of a trial where there is no stop signal presented, then the participant will have no interaction of the stop unit. So the beta go will not be interacting with the beta stop. So there will be no buddy sort of actually stopping the process. However, if there is a stop process, there is an interaction. So the beta go will interact with the beta stop, and that will sort of stop the process, and it will not let the going process reach the threshold theta. So those interaction parameters were the most key sort of parameters to focus on in the process of the augmentation. We do have model schematics in the further slides that will make this more clear. So this was the parameter variation 1. We call this the model 1. And we augmented the lambda parameters such that they actually affected only the part of the model of the interaction bit. So you can see that the mutual inhibitory interaction between the go unit, which is the disinhibition behavior, and the stop unit, which is the stopping behavior, those accumulators interaction, mutual inhibitory interaction was affected and or biased in a way with the parameter values of lambda stop and lambda go. So this was the first model. The second one, we tried to, because we were trying to rigorously test and see every single combination and see what was actually giving out the dynamics of the reaction time that we observed in the modified stop signal task, we tried to affect the mean threshold. So basically the growth of these accumulators towards the threshold and not the interaction in this case in the parameter variation 2. In the parameter variation 3, we tested and we tried to affect both the mutual inhibitory interaction as well as the mean growth rate. And these were the parameter constraints we had. The results led to this. This was the first architecture. So in here, you can see that the associative recall units are grayed out. So they're not playing a role and they're not biasing the processes. And this was the first parameter variation. Here they are green and they're affecting the mutual interaction. So you can see that there's a bigger purple circle in correspondence to the lambda go, which means that the go is actually in a way inhibiting the stopping process, and the stop process does not have a lot of impact on the go process. We had the parameters affecting the mean growth rate in this architecture. And we hoped to see the dynamics where the lambda go would have affected stop process more. And then we had both together. So the simulations ran for the first parameter variation in the parameter constraint settings, which are presented here, were the only model which actually led to the behavior that we were observing. The simulations resulted in the dynamics. So we had, this is a probability of signal response, which basically means the probability of responding on a stop signal trial was the highest for the first parameter variation model. And this was another comparison. This is only a subset of the simulations results, because there are a lot of other results, simulations of other dynamics that we looked at as well. But the red bar in here basically shows that the SSRT stands for the stop signal reaction time, and it was exactly the same. The simulations showed exactly similar ranges of reaction times for the first model augmentation. So in conclusion, we can say that we've actually been able to successfully build a model that showed the interaction between these two elements of inhibition behavior and associative memory. And in future directions, we are now currently working on, so we have collected some experimental data with a modified stop signal task in psychology paradigm. And we're now performing some fitting of this particular data with the first model for the future directions, which is what we're working on currently. Yeah. Thank you.
Thank you very much, Nilosmita. This was a nice topic. We are kind of running late for the next speaker due to the initial technical difficulties. So we don't have questions. I would pose you a few because it is kind of very much related to my research of interest. So I may email you, okay?
Yeah, sure, definitely.
And if any other person as well would like to email her, but we need to move on to the next speaker. Yeah. So if you can start sharing Xiqin LIU.